Paget disease of the vulva, extramammary Paget disease. Paget disease of the vulva, also known as extramammary Paget disease, EMPD, is a rare malignancy characterized by the intraepidermal proliferation of malignant cells. While it shares its name with Paget disease of the breast nipple, these conditions are distinct entities with different clinical courses and management strategies. In this essay, we will delve into the key aspects of Paget disease of the vulva, exploring its origin, clinical presentation, and diagnostic challenges. Intraepidermal proliferation of malignant cells. Paget disease of the vulva is characterized by the presence of malignant cells within the epidermis, forming Paget cells. These cells are large, with clear cytoplasm and prominent nuclei, and they often infiltrate the epidermis, leading to characteristic changes in the skin. The disease can affect various anatomical sites within the vulva, including the labia majora, labia minora, and perineum. Association with breast nipple Paget disease. While distinct from Paget disease of the breast nipple, Paget disease of the vulva shares similarities in terms of histological features. Both conditions involve the presence of Paget cells within the epidermis. Interestingly, these two manifestations are thought to arise from separate origins, despite their histological resemblance. Absence of underlying tumor. One notable characteristic of Paget disease of the vulva is its often sporadic occurrence without an associated underlying tumor. Unlike many malignancies, which originate from a primary tumor and later spread to surrounding tissues, Paget disease of the vulva may manifest without a clear underlying source. This poses a challenge in understanding its etiology and necessitates thorough diagnostic investigation. Origination from intraepidermal progenitor cells. The exact cellular origin of Paget cells and Paget disease of the vulva is not fully elucidated, but it is believed to arise from intraepidermal progenitor cells. These progenitor cells undergo malignant transformation, giving rise to Paget cells that populate the epidermis and contribute to the characteristic clinical features of the disease. Clinical presentation: red, scaly, crusted plaque. The clinical presentation of Paget disease of the vulva is distinctive. Affected areas often appear as persistent, erythematous, red, scaly, and crusted plaques. The lesions may be pruritic and mimic other benign dermatological conditions, leading to delays in diagnosis. Due to the nonspecific nature of these symptoms, Paget disease of the vulva is frequently misdiagnosed initially. Diagnostic challenges. Diagnosing Paget disease of the vulva poses challenges due to its rarity and similarity to benign skin conditions. A biopsy of the affected area is crucial for histological examination, revealing the presence of Paget cells within the epidermis. Immunohistochemistry may be employed to confirm the diagnosis, with Paget cells typically positive for cytokeratin 7 and carcinoembryonic antigen, CEA. In conclusion, Paget disease of the vulva is a rare malignancy characterized by intraepidermal proliferation of malignant cells, often presenting as a distinctive red, scaly, and crusted plaque. Its association with breast nipple Paget disease, absence of an underlying tumor, and origin from intraepidermal progenitor cells contribute to the complexity of its understanding and diagnosis. Given its rarity, a multidisciplinary approach involving dermatologists, gynecologists, and pathologists is essential for accurate diagnosis and management. Further research is needed to elucidate the pathogenesis of this condition and to develop targeted therapeutic strategies.